So many as a scientist started the study of as a mitochondria in as a different as a ways. Only as a for it is known as site for aerobic respiration, yeah, oxidative phosphorylation. But as time pass out, in as a past time, in past it was only called as sites for the ATP synthesis or through the process of oxidative phosphorylation. But if you look at also the other functions of the mitochondria, then mitochondria that provide precursors for at least synthesis of 12 amino acids. And this precursor mainly comes from the TCA cycle intermediates. The mitochondria that is also provides the intermediate for the synthesis of purine and pyrimidine nucleotide precursors. It is involved in synthesis of glucose or through the anaphylaxis reactions and otherwise. Third one, in eukaryotic cells, mitochondria is known as producers of the carbon dioxides. It produces carbon dioxides and also water. So that has the powerhouse of the cell. It is well known, another name of the mitochondria. But why it is called as powerhouse of the cell? Because it is act as a site for the organelle that has an well-developed electron transport system, a redox potential, where during oxidative processes, the electrons are produced, and these electrons is coupled also with the flow of the protons. So that has energy that is released during the oxidative processes in form of protons, and finally as energy of protons is captured by the enzyme ATP synthase that finally phosphorylate the ADP with inorganic phosphate to form ATP. Anyway, first of all, as the mitochondria that produces the ATP. What is as a functions of the a role of the ATP? If you look at the substrate, precursor, that is S, that is ADP with inorganic phosphate that enters mitochondria and as it produces ATP. ATP. What is the functions of the, what is the role of this ATP? If we look at this ATP that provide the energy for the synthesis of all kinds of the cellular functions. For you can also that is the membrane transport. That is the membrane transport, active transport, synthesis of protoplasm, including proteins, amino acids, nucleotide, nucleic acid, etc. If you look at the another side, then it has to provide the energy for locomotion, nerve impulse, cardiac functions, renal functions, reproductive functions, etc. So that all these kinds of processes that is carried out by different organs, all kinds of organs that need energy in vital energy that comes from the mitochondria in form of ATP. So that the ATP is known as energy currency of the cell. Whenever the energy, a cell has higher energy level, that has higher ATP level, it means there is reduced concentration of ADP at that time. This energy is utilized for the synthesis of other biomolecules, including fatty acids, cholesterol, fat, etc. And that fatty acids or fat or cholesterol that got deposited, that get deposited in different parts of the body. Sometimes the cholesterol is being utilized for the synthesis of plasma membrane, and cholesterol act as a precursor for the synthesis of all the steroid hormones in human beings. The cholesterol is also required for the synthesis of the biles. Second one, the architecture of the mitochondria. So then again, the outer membrane of the mitochondria is a continuous layer. There is no any modifications or injection in inner innervations takes place. But the inner membrane has finger-like indentation. That finger-like indentation is known as cristae. Our inner membrane that is mainly has to serve as a site for ATP synthesis through the process of oxidative phosphorylation. The chemistry of the outer membrane, if you look, then it has 40% lipid. And this is a lipid that has a comprises phosphatidyl inositol, outer membrane of the mitochondria that contain more phosphatidyl inositol, cholesterol, but less in another kind of lipids that is called as cardiolipin. And the cardiolipin that it has a lipid has a protein that cardiolipin that is a lipid that was first isolated from the cardiac muscles, and particularly the cardiac has muscles that perform severe inactive processes active time. It catalyzes nearly was 2020 more has a Krebs cycle once a time. Second role of the cardiolipin has that has makes permeability has for the certain proteins, certain substances, including protons, electron. However, inner mitochondrial membrane is impermeable, mostly impermeable for the charged particles. The inner membrane of the mitochondria has that contain has more has proteins, and this has a protein intercalate in between lipid molecules of the mitochondrial membrane. Another distinctions in between outer membrane and inner membrane on the basis of architecture is that is outer membrane has to contain has to 29 kd has to protein. 29 kd has to protein. This protein has isolated as using different centrifugation techniques. Later it is separated on the electrolysis band. So this 29 has to kd protein that is called as porine, or it lines the porine, and as a result of the presence of the 29 kd protein, the outer membrane is permeable for the non-polar molecules, also for oxygens and other gases. So the 29 kd that is a specific protein that is present in the outer membrane, and it remains absent in the inner membrane. Now other architecture of the mitochondria that is known for presence of enzyme, and the membrane, outer membrane, inner membrane, intermembrane space that has a specific enzyme. If you look at the outer membrane, intermembrane space, inner membrane, matrix, etc., all are specific for containing the another kind of enzymes, so that the enzyme is not repeated in any kind of mitochondrial chamber. We are trying to represent, for example, this is the outer membrane, and the very first enzyme I am writing here, it acts as a marker enzyme. The marker enzyme that is known to presence of the occurrence of the enzyme in a particular site. Whenever this enzyme, if you separate the mitochondria using different techniques, then the enzyme that remain associated with any membrane, then you can say this membrane is outer membrane. The very first enzyme, a marker enzyme of the outer membrane, that is monoamino oxidase. Monoamino oxidase. So this is the enzymes that is present at the outer membrane. Second the protein that is present in outer membrane, that is the cytochrome B5. Third most important enzyme that is known as rotinone NADP insensitive oxidase. The rotinone, that is a kind of insecticides. One question that is, because of the the agriculture farmers using the insecticide to protect the crop from the insect, and then how insecticide also works also there, because animals, yeah, the insects are also eukaryotic organism, and plant are had also eukaryotic organism. So why has the animal insect has to get has to died or remain away from the as a plant, and as a plant has to remain unaffected. So because as if the rotinone and other insecticide, if you apply on the agriculture as a crop, then, for example, the rotinone, whenever act upon the as a plant cells, then the plant mitochondria has alternate electron NADH systems. If he has because as a rotinone that inhibit the complex first of the mitochondria, so that is the electron transfer from the substrate to electron carriers, very first as a complex NADHQ reductase. The NADHQ reductase that contain the iron, and as from the iron sulfur, the electron is transferred from the iron sulfur to the quinone, so that in case of the insect or human beings, animal as a cell, the rotinone prevent the transfer of electrons from the iron sulfur proteins of the complex first to the UV quinone, so that electron does not flow from complex as a first to the UV quinone. But in case of mitochondria, they have a plant mitochondria has an alternate NAD dehydrogenase, so that if insecticide is applied as so there, then during oxidative aerobic respirations, the electron is directly has to transfer without passing to the complex as a first, and electron is directly passed to the UV quinone, so that the insecticides is applying on the crop plants that is so doesn't has to harm that's a crop, but as the same insecticide it is applied in case of the animal cell, that then an animal cell that is that block the respiratory chain of in mitochondria. That's why the second enzyme, a most important enzyme that is present in the outer membrane of the mitochondria, that is the rotinone. एक क्वेश्चन आता है नेट में, आप ध्यान रखेंगे इसमें, rotinone insensitive NADH dehydrogenase या reductase, not dehydrogenase, actually it is the reductase enzyme. Third in case of that is the fatty acyl coenzyme synthetase. It is the enzyme so that is used as for the during the oxidations of the fatty acids, because very first reactions of the fatty acid during their oxidations takes place in cytosolic sites, and so but rest of the reactions takes place in matrix of the mitochondria, but so the mitochondria does not allow the transport of the coenzyme SH. That's why the enzyme has to very first catalyze the oxidation activations of the fatty acid in presence of enzyme fatty acyl coenzyme synthetase. Second has to the outer membrane that also has carnitine transporter system. You will also find as a letter. Then also the outer membrane of the mitochondria that contain monoamino oxidase, cytochrome B5, and most important insecticidal has enzyme for in sensitive insecticidal enzyme in mitochondria, in particularly in case of the plant cell, that is the rotinone insensitive NADH reductase. Third one, the fatty acid synthetase, the fatty acyl coenzyme synthetase. Second, as if you look at the intermembrane space, then intermembrane space, so that is the very first marker enzyme, that is adenylate kinase. Adenylate kinase, it is a marker enzyme of the intermembrane space, not inner. Intermembrane space, followed as by nucleoside diphosphate kinase. Second enzyme that is present in the inner membrane space, the intermembrane space, that is the nucleoside 5' diphosphate kinase. And other as a DNA H first, and as a phosphatase, etc. This is also present in the intermembrane space of the mitochondria. But now has to be come to the intermembranous matrix. And so the matrix that has contained a lot of the enzymes. Lot of the enzymes, that is very first marker enzyme, that is the citrate synthase and isocitrate dehydrogenase. This is two enzymes together act as a marker enzyme in the matrix. Very first enzyme citrate, synthetase and isocitrate dehydrogenase, followed by another enzyme like aconitase, alpha ketoglutarate dehydrogenase, pyruvate dehydrogenase complex, alpha dehydrogenase complex, succinate dehydrogenase synthase, that catalyzes the synthesis of succinyl coenzyme from the pyruvate. One question is here in NET has two or more times. So one question was asked, which enzyme of the Krebs cycle that is present on the inner membrane of mitochondria. The entire enzyme of the Krebs cycle that is present in the matrix of the mitochondria, but one exception is, that is the succinate dehydrogenase. Succinate dehydrogenase, it is the enzyme that is present on the inner membrane of the mitochondria. So that one question is here. The malate dehydrogenase, isocitrate dehydrogenase, citrate synthetase, alpha ketoglutarate dehydrogenase, succinyl coenzyme synthetase or succinyl coenzyme kinase, fumarase, and so the enzymes of the fatty acid synthesis, fatty acid oxidations like enol coenzyme dehydrogenase, thiolase, thiokinase, or the one has more enzyme, that is the acyl coenzyme dehydrogenase. This is the enzyme has present in the matrix of the mitochondria. Another question, the mitochondria that also contain the genetic as a component. Genetic component, that is the DNA. And as a DNA of the mitochondria, however, if you look at the basic compositions of the human mitochondria is, the human mitochondria has a DNA that is the circular, covalently close circular DNA. The number of the base present in the has of the human DNA is nearly about 16,502 base pair. So that mitochondrial DNA is composed of 16,502 has a base pair. That's why the mitochondrial has a DNA that is self-replicable, but however its enzyme, replicating enzyme has comes from cytosol that is synthesized on the nuclear has a DNA. So that as the mitochondria is interdependent upon the cytosol and nucleus for enzymes, for other functions, metabolic functions that is carried in the mitochondria, that is imported from the cytosol, and the protein and enzyme that is synthesized on the nucleus. However, mitochondrial has a DNA that contain nearly about 37 genes. 37 genes. This has out of the 37 has genes, the 13 has genes. The 13 genes are involved. You have synthesized the proteins for complex first and complex second or complex third. However, complex third is not being synthesized. But the complex first, complex third, and fourth. And the polypeptides, few polypeptides synthesized on the mitochondrial gene. Entire the respiratory chain complex protein is not being synthesized on the mitochondrial DNA, but as a mitochondrial DNA has 37 genes. Out of 37 genes, 13 genes encode for the protein that makes the respiratory chain. Secondly, rest of the 24 genes, the 24 gene that synthesize the RNA through the process of transcription, and this RNA mostly belong to transfer RNA through via messenger RNA. That is it encode the transfer RNA for the assembly of amino acids during the protein synthesis. However, another distinction in mitochondrial matrix, that is the presence of ribosome. And the ribosome of the mitochondria differs from prokaryotic and eukaryotic ribosome, because prokaryotes has two cells of 70s consisting of both subunit 70s, and in case of mitochondria, that is in case of eukaryote, that is the 80s. However, eukaryotic cell has two or more kinds of the ribosome, organelle ribosome, three cytosolic ribosome, membrane attached and membrane bound as a ribosome, in which also the chloroplast has 70s, but mitochondria has 55s, 55s subunit. The smaller subunit that consists of the 25 subunit, whereas a larger subunit that consists of the 35 subunits. And this has a 25S subunit, it has to contain the 12S ribosomal RNA, 12S ribosomal RNA. 12S ribosomal RNA. Whereas as a 35S, it contains 16S ribosomal RNA, 16S. So that it also gives evidences, evolutions of the mitochondria, evolutions of the organelle or eukaryotes from the prokaryote, because the 16S ribosomal RNA is the characteristic features of the smaller subunit of the prokaryotic ribosome. So that another has a distinction, and that is the ribosome. It differs from other has some cellular ribosomes. So that matrix of the mitochondria that has cations, ADP nucleotides, ATP, GTP, CTP, TTP, UTP, so that all kinds of the nucleotides are as a present, consisting of both kinds of ribonucleotides as well as deoxyribonucleotides. Anyhow, the numbers of the ribonucleotide always remain higher in any cases, even in the nucleus also. So that mitochondria also has higher number of ribonucleotides, not only as for the transcription processes, but also that energy capturing. Third has the enzymes present on the inner membrane. And the very first marker enzyme of the inner membrane, that is the electron transport system. Respiratory has a chain, ATP synthase, carnitine transport system, carnitine palmitoyl acyl transferase, translocase, transferase first, transferase as a second, succinate dehydrogenase, and electron transport system that consists of the 4 complexes: complex first, complex second, complex third, and complex SO4. The complex first is known as NADHQ reductase. Complex second is known as succinate Q reductase. Complex third is known as cytochrome C reductase, and complex 4 is known as cytochrome C oxidase. If you just look at out of 4 complexes that is present in the inner membrane, on the inner membrane of mitochondria, the 3 complexes are concerned with reductase, and only 1 has the complexes that is known for the oxidase enzymes. In another has the 4 complexes. One has the versatile component that is present in inner membrane as well as in outer membrane, or as it is the known as mobile particles, that is known as Q, ubiquinone. Sometimes it is called as Q particles, ubiquinone particles, ubiquinone. This ubiquinone, and it has received the electrons from the complex first and complex second, and but it transfer electrons and protons to the complex third. So that inner membrane of the mitochondria that has a specific enzyme, and most of the enzymes are known for electron transport system, but has the 2 enzyme to act, another enzyme that is succinate dehydrogenase, and second one is the carnitine transporter, carnitine shuttle, that consists of at least 3 enzymes. Very first, that is the transferase first, translocase, and transferase third. The transferase first is attached with the fatty acid that has to enter the matrix of the mitochondria from the cytosol. For example, if you have to take palmitic acid, then the name of the enzyme will be palmitoyl coenzyme transferase first, carnitine palmitoyl translocase, and palmitoyl transferase as second. Similarly, if you have stearic acid, asteroyl coenzyme carnitine acid asteroyl coenzyme transferase first, then translocase and transferase third. Transferase second. So that is the enzyme of the carnitine transporter, that depends upon the fatty acids that has to transport. Because the mitochondria has that allows the freely translocations of 4 carbon compound, yeah, 8 carbon compound, yeah, 12 carbon compound readily. But as the fatty acids that contain 16 carbon onwards, so then and it has enters the mitochondria through the subtle system, a carnitine transport system. Second another objective is that the mitochondria, yeah, in any living system from bacteria, yeah, so the organism has that has mitochondria. The mitochondria has that allows the oxidations of fatty acids up to 18 carbon. But however, as we consume as the fatty acids more than as 20 carbon. For example, in winter season, the peoples of the temperate regions that also consume groundnut more. And groundnut also that contains arachidonic acid, that is the 20 has carbon. However, this 20 carbon has arachidonic acid does not also utilize as a fuel for the synthesis of vital energy like ATP, rather than they also undergo oxidative processes during which the protons are dissipated, the energy is dissipated in form of heat. That's why as during winter season, as we can all consume as the groundnut for heat production and other precursor also, but it is not as utilized as precursor for the ATP synthesis. So that mitochondria of any higher or eukaryotic organism that oxidizes up to 18 carbon fatty acid, including both saturated fatty acid as well as unsaturated fatty acids. Okay. So that a basic, and also the another also that contain also the inorganic phosphate or inorganic pyrophosphate. Most important, another enzyme also that is present in the matrix, that is the inorganic pyrophosphatase enzyme, and also coenzyme A, that is the vitamin B5, and coenzyme A is the B5 that act as a carrier also of acyl group, acetyl has a group, succinyl has a group, etc. Even though so that it act as a carrier and it contains the coenzyme acid sulfur hydryl group, so that during which oxidation reduction processes that has the enzyme, yeah, carrier molecule that contain the sulfur hydryl. This sulfur hydryl comes from the cysteine amino acids. That's what we will get. The basic architecture of the mitochondria is like that the enzyme system mitochondria is separate, lipid component of the mitochondria is separate, the organelle has that contain has DNA, RNA, मैंने ट्रांसफर आरएनए. हाँ, दूसरा क्वेश्चन इसमें और आता है कि बिकॉज ऑफ माइटोकॉन्ड्रिया that has nearly what has the transfer RNA that can incorporate in protein synthesis of the mitochondrial as a protein, that is only the 18 amino acids, so that they have only have 18 kinds of the transfer RNA, 2 has नहीं. और दूसरा जो ऑब्जेक्टिव है, बिकॉज इन जेनेटिक टर्मिनेशन कोडोन UAA that act as a termination codon during the cell protein synthesis in cytosols. But however, in case of the mitochondria, this UAA termination codon that incorporate the phenylalanine during the protein synthesis, so that it put the universality of the genetic code. That is another objective. But anyway, so this is the basic architecture of the mitochondria. Up complex, first complex, second complex, third and complex 4, I am coming one to one. Then it later one, we will get functions. Or one component in the mitochondrial membrane has that remains left, that is the subtle system. Subtle system has because the inner membrane of the mitochondria does not allow the easy transport of the charged particles. For example, the oxaloacetate, and the oxaloacetate, that is the act as a very first precursor for the synthesis, condensation of the acetyl coenzyme to synthesize and to produce the citric acid, citrate. And so whenever energy reach your mitochondria is full of the ATP, the then has a citrate that act as a precursor for the synthesis of fatty acids. So the citrate splits in the mitochondria in presence of citrate liase, but this citrate liase has split the citrate into oxaloacetate and acyl coenzyme. but this auxiliary state does not leave the acyl coenzyme does not leave the mitochondria for the synthesis of fatty acids, so that it comes outside through a shuttle known as citrate shuttle. Another the shuttle system that is the malate aspartate shuttle. Malate aspartate shuttle, it is well-known shuttle that is the link that act as a connecting link in between glycolysis and TCA cycle. Pyruvate is another the end product of the glycolysis, but pyruvate that is the anions that does not enter the mitochondria directly through the inner membrane, but inner membrane of the mitochondria has symport system, and through the symport the pyruvate enters along with the proton, because pyruvate carries one negative charge and proton carries one positive charge, so that both has a charge balanced to each other. That's why pyruvate enters the matrix of the mitochondria along with the proton, because inner membrane of mitochondria has to maintain two objectives. One objective that it has to maintain the electrical gradient and chemical gradient. Chemical gradient that is known for pH. And any active respiring mitochondria that does not, it means active respiring mitochondria means they are actively involved in the oxidations of the foodstuff that produces both carbon dioxide as well as reducing equivalent, that is the 2H. And this has a 2H that contain 2 electrons and two protons. And so that whenever concentration of proton increases, then pH of that area also decreases, so that there is chances to make the medium acidic. But however, in any actively respiring mitochondria, if proton concentration increase, but however does not decrease the pH beyond the 6 point something, it means किसी भी केस में 6 से pH कम नहीं होगा, the 6.4, 6.5, or the pH never increase beyond the 7, so that it doesn't maintain the alkaline pH. And this has a proton gradient that has to make the proton concentration, and it is the precursor source for the proton pump, that is for the ATP synthesis. One. Now we come on the topic compositions of the respiratory complex first. The complex first is known as the largest complex of the mitochondria. And the entire respiratory has a complex that constitute nearly 15% of the total protein of the inner membrane of the mitochondria. And the complex has first that contains the 15, it consists of the 15 polypeptide chain with 6 iron sulfur protein. 6 iron sulfur protein. It also contains FMN and FD. Or it accepts the electrons, the reducing equivalent from the enzyme that it causes oxidations of the foodstuff, that is linked with NAD positive. NAD has to link to dehydrogenase enzyme. During glycolysis, glycerol 3-phosphate dehydrogenase. In case of the Krebs cycle, the 3 has to dehydrogenase enzyme is linked with the NAD, that is the isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, and malate dehydrogenase. Another enzyme has a pyruvate dehydrogenase complex that is also linked with NAD. It is the NAD linked enzyme. So that has the NAD has the nicotinamide adenine dinucleotide. It accepts 2 electrons. So that if has a 2H, if you split the 2H, then it has the split into hydride ion and also the proton ion. If this is the proton ion, this is the hydride ion. This is the hydride ion. Also that it carries 1 negative charge. So that it contains 2 electrons and 1 has a proton, where it contains only 1 proton. So that as it is, due to the presence of 2 electrons, it has bears 1 negative charge, so that it has accepted by NAD and it is reduced into NADH. Some books have written and some NADH2, but it is totally wrong, because NAD accepts 2 electrons and 1 protons. Whereas in case of the FAD, that accepts the 2 electrons and 2 protons together, but NAD is not. NAD accept only 2 electrons and 1 proton. So this NAD linked has to NAD. That's why the complex first, that is known as NADH Q reductase, that consists of 15 subunit and 15 polypeptide subunit with 6 iron sulfur protein. It also contains the FMN and FAD and NAD. It accepts the electrons from those oxidative processes that is catalyzed by the NAD-linked dehydrogenase enzyme. And this enzyme is also sensitive to, just as we have told, rotenone and other insecticides that has to inhibit the transfer of the electron, particularly from the iron sulfur protein to the ubiquinone. And the NAD complex first is coupled for the flow of both electrons as well as protons. It has to transfer electron to the ubiquinone, that is the versatile protein, ubiquinone, and UB. This ubiquinone, and this also then it reduces finally into QH2. If it accepts as both electron and proton, then it is reduced into QH2. That is the quinol as a molecule, hydroquinol banata. It is known as hydroquinol. And also the complex first is also coupled with the uphill transport of the protons. So that as the proton has that is its concentration increases in matrix of the mitochondria. So then as concentration of proton increases in matrix of the mitochondria, so that this proton gradient develops a force known as proton motive force. This as a proton has enters the inner membrane space. So that's why there is also electrochemical gradient. And as a proton accumulates in the inner membrane space, then pH of the matrix of the mitochondria increases, and also the membrane potential that is changed nearly 1.14 potential. There change in the pH nearly 1.4, yeah, 1.14. Some books have written that in change in the pH. And later this proton accumulates in the inner membrane space. The latter it is transfer to the ATP synthase. Later one in you will find in complex 4. That's why NAD is concerned with that. Second one has to transfer electron to the ubiquinone, and quinone has to get reduced in 2 steps. Very first semi-quinone, or second one whenever one more has electron, the proton accept, then it change into the hydroquinol, a dihydroquinol. Now has a complex second. Complex second mostly consists of the 2 polypeptide chain and has 1 or 2 iron sulfur protein. Another one, it has to contain the FAD. So that this enzyme is also sensitive that catalyzes the oxidation reduction reaction, particularly at the that is concerned with the enzymes that catalyzes the oxidation of a fuel, a substrate that is linked with the FAD. So that is during oxidative processes in TCA cycle, the succinate dehydrogenase FAD link and acyl coenzyme dehydrogenase during the fatty acid oxidations, it is the FAD has a link. And FAD has that contains the riboflavin, and it is the riboflavin, it is vitamin B2. And so this vitamin has B2, however, NAD, that is the vitamin B3. And as vitamin B3 that contains 2 kinds of nucleo NAD. One is the NAD, another with the NADP. There is distinction in between NAD positive and NADP positive. The NAD has a positive which directly has a relationship. The reactions are linked with the reactions that had oxidation process, oxidative process. But NADP is more abundant in case of the chloroplast or as a cell that is involved for the synthetic process. So that NADP is known for reductive process, NADP positive, that is involved in reductive as a process. That is the reductive means synthesis. चाहे न्यूक्लियोटाइड का सिंथेसिस होना है, चाहे आपका फोटोसिंथेसिस के समय ग्लूकोज का बनना है. That's why NADP is concerned with the reductive as a process, whereas NAD positive that is concerned with oxidative process. That this FAD has to accept both 2 electron as well as 2 protons, so that it is reduced into FADH2. After accepting, then it change into the FADH2. And this FADH2 transfer directly has electrons to the ubiquinone, so that if as an electron moves on the inner membrane of mitochondria from complex 2 to ubiquinone and then complex 3 and complex 4, then it is able to produce 2 molecules of ATP rather than as X electrons enters the complex first via NADH dehydrogenase, NADH-Q reductase, then it has to transfer the electron to ubiquinone, then to complex third, and then complex 4. That's why it generates generate the energy potential that is equivalent to synthesizing a phosphorylating 3 molecules of ATP, etc. Now has a complex third. And complex third that is known as cytochrome C reductase. So that it contains cytochrome B1, cytochrome C. Yeah, sometimes cytochrome B, cytochrome B1, cytochrome C. It expands the mitochondrial membrane and as it contains the copper. No doubt, also contain the iron sulfur protein, and as along with the iron sulfur protein, it also contains copper. So that is the copper has a present that undergoes oxidation and reduction. Yeah, so that is the copper has that is when cupric has a form and cuprous has a form, and iron also has that is the ferrous and as a ferric. Depends upon the oxidative process reactions during precursor. Then all the 3 electrons are removed. Then by ferric has configurations, and so then it had chances for insertion of 1 more proto electrons. Then it is the ferrous configuration, so ferrous form. So that the complex third that contain the copper. So that there is 2 metal ion, and both the metal ions present in the complex third. And as the complex third that accept the electrons and protons from the ubiquinone. So that ubiquinone has to get oxidized and it transfer its electrons and protons to the cytochrome B1, then to B cytochrome C, etc. And it also proton has a pump system. And that's why it also able to capture the energy of this proton, and that is equivalent to phosphorylate the ADP with inorganic phosphate and form the ATP. So that only ATP is not being synthesized as whenever in complex second, because as a complex second does not have the proton channels. That's where one question comes in case of the complex 2. Why complex 2 is not ready for the synthesis of ATP? Because a complex second that has not proton channel. The proton channel is present in complex first, complex third, and complex 4. Last one that is the complex 4. The complex 4, it is known as the larger complex. Largest complex that is the complex first. Complex second is the smaller subunit, whereas complex third is the large, and as a complex 4 is the larger as the complex of the mitochondria. It contains cytochrome A and cytochrome A3. And cytochrome A and cytochrome A3 that also contain iron, copper, along with the iron. So that it is the copper. And two questions has remained more. That is the commonly known as a brown fat. Brown fat should not be confused as to it with the color. But the brown fat is present in the adipocytes of the newly born mammals. In both wildlife, even though humans. And the newly born mammal baby that has contain large amount of the brown fat. And brown fat is nothing but it is the mitochondria that is present in the adipocytes. So that the numbers of the mitochondria in the adipocytes is higher and higher, and as higher and higher, so that presence of the copper this seems to appear as a brown. That's why it is known as a brown fat. It is present in the newly born baby, particularly in the mammalian baby. And whenever you take the baby, you touch the baby, then you feel some burn, because your newly born mammals, they survive in open natural environment where there is no any system to protect during the cold environment. So because they have higher number of the brown fat, and brown fat because the proton has a flow during the oxidative phosphorylation reactions directly as a transfer to the complex SO4 that catalyzes the oxidations of the electron with the oxygen to form H2O, and as the rest of the SO proton that is in another SO case that is captured for the ATP synthesis, but in case of the SO brown fat adipocytes of newborn SO baby, mammalian SO baby, the energy of the proton dissipates in form of the heat, so that heat is generated rather than ATP is produced in SO adipocytes that contain the brown SO. So that is the complex SO4 is the larger complex that contain the 6 or more polypeptide chain along with two molecules of the copper, and it contains the oxygens, and that except that transfer the oxygens that transfer the electrons directly to the oxygen to form the water. And one has that is the elementary particle that is present on the inner membrane of the mitochondria. And if you have an elementary particle has that, if you look at this, that consists of the three subunits that is known as head. That is also known as connecting link, yes, top, and also that is the base. It is known as proton channel. This is the stalk, and so this is the knob. It contains the ATP synthase, and so the proton has the proton channel that is mostly in hydrophobic in the nature, so that it remain embedded in the inner membrane as a membrane of the mitochondria. That also due to the nature of the hydrophobic nature, it remain embedded, and it act as a passage for the flow of proton from the inner membrane space. So that is the inner, if you look at the elementary particle, then its location just like as this. Just so that the base of this proton channel as that remain as buried as in the inner membrane as a lipid. So that is the proton that has moves from matrix during the oxidation reaction and accumulates into the inner membrane space. Later one during the ATP synthesis, it transfer and it to the proton has a channel.
देखकर वो हील होने लगा है तो वो भी अपने प्यार का इजहार कर देता है और दोनों किस करने लगते हैं, लेकिन तभी वहां पर हलचल।
बेटा क्या आ रहा है? Then the head and knob that is called as F1. This is known as F1 particles, and this is known as F0. F0 and entire has this subunit. This is the question is because it is sensitive to a kind of antibiotic that is known as oligomycin. So that has the stock of the elementary particle that is known as oligomycin sensitivity conferring protein, that is most commonly known as OSCP. And this OSCP, whenever the mitochondria is treated with the oligomycin or other proton blocker, so then also it has to block the protons from the movement of the proton channel to the ATP synthase. Because this ATP synthase that contain the ATP synthase enzyme, and this ATP synthase, later one I am coming one by one. So that entire enzyme that consists of the 9 subunits of 3 kinds, alpha has 3, and also beta has 3, alpha 3 and beta 3, and this has 3 enzymes that consist of the gamma delta epsilon. And this gamma delta epsilon is that make the hydrophobic nature of the proton channel or a stock of the elementary particles. So that is the alpha 3 that is mostly hydrophobic in nature, and however if you take the beta 3, this beta 3 subunit that consists of 3 configurations. In one configuration, the beta 3 consists that consists so the beta empty. This is the empty. Empty means it can accept the ADP or ADP. Second one is the beta has that contain the ADP. ADP or beta has a 3. यहाँ पर that is also it contains another as enzyme. Yeah, the minor membrane of mitochondria that contain another enzyme that is known as ADP ATP translocase. ADP or ATP translocase. ADP ATP translocase, it means the cytosol that contain the ADP after utilization of its energy, then ATP convert into ADP, then ATP must be imported into the mitochondria. And secondly, the mitochondria so that has this enzyme that phosphorate the ADP with an organic phosphate to form the ATP, so that ATP must be comes in the cytosol through a transport system, a translocase system. So this is mediated by a carried out by an enzyme known as ADP, ATP or ATP, ADP translocase enzyme. Its energy that later one we will explain later more. So this also the ADP, and third one also that has the catalytic sites that has catalyzed the synthesis of ADP with inorganic as a phosphate. Sorry. Inorganic phosphate. So that the beta subunit whenever and also the beta 3 just beta 3 subunit or one subunit that the beta empty is means that has no, but if the site has to accept the whenever also the ADP comes in contact with the beta subunit, then as a beta empty accept this ADP, and then it get converted transformed into beta ADP, or as whenever a phosphorylation of ADP with inorganic phosphate takes place, then as ADP get phosphorylated and form the ATP, and as this ATP after as its formations, then as the another has to submit get as a free and as a ATP is that is released into the matrix. So that is the ATP after formations of the ATP and it releases into matrix, and then it again become the empty. Very first that is the empty. Then as ADP has a bounce that it that undergoes conformational changes. After conformational also changes, the ADP phosphorylate with the inorganic phosphate and form the ATP. Whenever ATP is formed, then as ATP released, then as it becomes beta empty. So that is the conformations of the beta subunit, that is the rarely has to undergo whenever ADP, ATP or oxidative phosphorylation is vigorous. The gamma delta epsilon that is more hydrophobic that remain into the inner membrane of the mitochondrial membrane. So that is the elementary has a particle, yeah, fundamental has a particle that is present in the mitochondria, also of the mesosome, that has of the same as a protein structure that consists of the three component, the F1 particle, F0 particle, and also the stop. Or F1 as that contain as the in the catalytic subunit known as alpha 3 and beta 3. Alpha 3 stabilizes the 3 subunit of the alpha proteins, stabilizes the ATP synthase enzyme, and beta 3. Yeah, one question comes from that: which of the following subunit of the ATP synthase has catalytic sites or catalytic activity? So then it is the beta has a 3 that has catalytic activity that is related with ATP synthesis. So that basic structure of the mitochondria, just like as of that. The question also comes from the mitochondria, so that is the one architecture in case of cardiolipin, and also the marker enzymes. And now, any question, if you any have students, you have any query as of for the architecture of the mitochondria, then you can ask personally or as. So then architecture is now over. now as we have two functions of the mitochondria. So the functions of the mitochondria, the very first function that is known as oxidative metabolism. And so that all kinds of the monomeric subunit like as a glucose, amino acids, and fatty acids are finally or ultimately oxidized in form of CO2 and H2O with concomitant productions of energy in form of ATP. But the one component function that is the ATP generation, that's why I will come later. Second, mitochondria synthesizes all the monomeric subunit through the intermediates. Just for example, in nearly about more than 12 amino acids, which precursors comes from the mitochondria. If you example as a take, like as oxaloacetate, this is the oxaloacetate. And whenever oxaloacetate that accept the amino group, then it change into the aspartate. Or this aspartate has to further accept the amino group during the transamination process. It has to synthesize the aspargine. Similarly, the pyruvate, if as a amino transamination pyruvate undergoes the transamination reactions, then it change into the alanine. Similarly, alpha ketoglutarate. Alpha ketoglutarate has, if it is the transaminated, then it synthesizes the glutamate, and glutamate is further transaminated and it get the amine groups, then it synthesizes the glutamine. And so glutamine has, so that is a well-known amino acid for particularly for the amino group donor for the nitrogenous synthesis of nitrogenous base, both urine as well as pyrimidine. So that's why it's the same. You can see leucine, isoleucine, and the phenylalanine, tyrosine. These amino acids, nearly about 12 or more amino acids are synthesized from the intermediates of the TCA cycle. The very first, that is the oxidative process. I will see catabolic process. I will already know because number of amino acids that has two skeleton, carbon skeleton and nitrogen skeleton. And there's the nitrogen skeleton, that is the amino group NH2 or NH3, and rest of the carbon skeleton like as a hydrocarbon and carboxylic acid group. So that carbon skeleton of the amino acids that enters the matrix of the mitochondria for further oxidations through the TCA cycles in form of CO2 and H2O, and during that process ATP generated. The nitrogen skeleton that is changed into NH2 or NH3 and finally into ammonium ion NH4 that is more toxic to the bodies. That's why it must be removed. And so that has along with the blood vessel, it enters the liver cells, hepatocyte cells, and hepatocyte mitochondria that serve as a center for very first reaction of the ornithine cycle. So that formation of carboamide phosphate during that takes place in mitochondria. That's why it also accepts the ammonium ion. And later one is synthesizing the ornithine, and ornithine has a further and change into so caramel phosphate, then change into citrulline, citrulline arginosuccinate, arginosuccinate arginine, or arginine splits into the ornithine and urea. So that urea has to get rid of. So that very first reaction, mitochondria, hepatic mitochondria that act as a connecting link, because very first or two more reactions takes place in matrix of the mitochondria, and rest of the reactions takes place in the cytosol. So that the formation of the urea in higher vertebrates, ureotelic as organism that in need has mitochondria as well as cytoplasm. So that second function, first function energy, second function that is the synthesis of synthetic process, the urea synthesis, amino acid synthesis, fatty acid synthesis. So that as the fatty acid synthesis, which precursor is the acyl coenzyme. So that acyl coenzyme has that is formed after the oxidations of the pyruvate that takes place in matrix of the mitochondria that must be transported in the cytosol for the synthesis of the fatty acid. So that mitochondria as act as a major only has a sources for the acyl coenzyme that is transported from matrix of the mitochondria to the cytosol. So that another has a synthetic has a process. Third has that is the anapleuritic reaction. One is the known as anapleuritic reaction. anapyretic reaction, because whenever glucose blood sugar level depletes, as we have as a fasting or fasting, during as of fasting, as a glucose as level downs and down, that the neuron needs the glucose for its functions. So then as whenever blood glucose has to deplete, then other food sources like fatty acids or intermediates of Krebs cycle, mostly oxaloacetate. So this oxaloacetate, this also and get reduced with the NADH plus HIL and change into the malate. Then as mitochondria has malate as a transporter system, so that malate enters the cytosol from the matrix of the mitochondria, and this as a malate as a further oxidizes into oxaloacetate, and oxaloacetate substitute then NADH plus HIM. This oxaloacetate undergoes decarboxylation reaction and one molecule of carbon dioxide is released and form the pyruvate. And this is a pyruvate that is the end product of the glycolysis, but however, it is the initial as a substrate as a for the gluconeogenesis. So that mitochondria has to generate the initial precursor as a for the synthesis of the glucose that comes from the mitochondria. So that synthesis, the synthesis, it synthesizes the glucose, it synthesizes the amino acid, it produces the precursor for the fatty acid biosynthesis, cholesterol biosynthesis, fat acid molecules. Third one is it produces the precursor for the synthesis of precursors of the nitrogenous base like purine and also pyrimidine. It also holds out the ammonium ion for the synthesis of urea. So that is the mitochondria. Another, that is the mitochondria that contain the DNA or RNA. That's why it also synthesizes the protein, synthesizes the messenger RNA, transfer RNA, and also the messenger RNA that contains the codon has for the information for the synthesis of proteins, and also the ribosomal RNA mostly comes from the cytosol. That's why it synthesizes RNA, DNA. So the synthetic process of the mitochondria is that also. Or second, already because in natural balance, carbon dioxide is also required for the photosynthesis, so that community respirations, during community respirations, the CO2 is exported from the mitochondria and for the photosynthetic processes. That's why it is the producers of carbon dioxide, it is the producers of the H2O water also. Or it is the consumer of the oxygen. If as a consumer, oxygen is not consumed, particularly when we have fasting, or during as a fasting still has to be breathing, then concentration of oxygen in our as a body has to takes place. The during as a which peroxidation reactions takes place during that as oxygen, mostly the endoplasmic reticulum and lysosome that is a product initiate the engulfing of the mitochondria, particularly lysosome. Begins the engulfing of the mitochondria, so that is the higher that mitochondria contain the oxygen due to the autophagy nature of the lysosome. It engulf the itself as a mitochondria of the same cell in which lysosome is present. So that during that has a time lysosome protect the cell from the after the peroxidation reactions also. Main has a function that is the energy. Sir, we can continue more. Madam. Anyone? आवाज नहीं आ रही बेटा मेरा?
यस सर, आ रही है आपकी आवाज।
आ रही है। क्या नाम बेटा मैडम आपका? क्या नाम आपका?
सर महकनाज।
तो महकनाज है ना, और कोई नहीं है? गीतिका नहीं है?
गीतिका भी है सर।
कहाँ है? बोल नहीं रही है।
कनेक्ट तो है सर।
अच्छा, anyone has to now I conclude my lecture today.